Today, we are setting aside the world of pure software. We're going somewhere different.
A lot different.
Yeah, we're diving into a quiet, but I think truly revolutionary space. I want you to picture a lab bench where the computational frontier isn't marked by, these blazing heat sinks and loud fans,
Right.
but by the gentle hum of incubators.
Yeah.
This is the place where living cells are actually meeting logic gates.
It's where biological circuitry for the first time is forming a real two-way connection with silicon hardware. A genuine conversation.
And this isn't science fiction anymore, is it? We are exploring the creation of these hybrid computing systems.
That's right.
It's forming a literal bridge where dynamic, evolving biological networks, and researchers have this great term for it, wetware.
Wetware, yeah.
It engages in a functional duet with precise, fixed digital logic. So our mission today is a really extensive deep dive into the technology, the architecture, and of course the immense challenges of something called organoid intelligence.
It's a heavy topic.
It is. It's complex material sitting right at the intersection of neuroscience, bioengineering, and computer science. But by the time we're done, we will have provided you with a thorough shortcut to being truly well-informed on a development that could, it could fundamentally redefine how we think about intelligent systems.
And that redefinition, I think, is crucial because the stakes are already incredibly high. We're not building these hybrid systems just out of scientific curiosity.
Not just a what if.
Not at all. We are doing it because our current purely silicon-based computational model is hitting some fundamental physical and economic limits.
We're hitting a wall.
We're running into major walls. The first is extreme energy intensity, both for training and deployment. Then you have the inherent brittleness of AI models.
Right, they break easily.
They do. And there's this frustrating inability of even our most powerful chips to generalize, to adapt seamlessly when the conditions on the ground shift. And the source material makes a really crucial distinction here. This is not just another sophisticated software simulation of life running on a conventional server. It's the complex, the energy efficient, the inherently plastic ability of life integrated directly into the computational stack.
Okay, let's unpack this. We have to start right at the beginning. What exactly is the biological raw material we are plugging into the digital world? I mean, to even begin this discussion, we need that foundational component, the living tissue. And the necessary precursors are what researchers call brain organoids.
Right.
Now, for listeners who may have only heard that term in passing, these aren't full conscious brains in a jar, but they are absolutely essential to this whole endeavor.
They're the building blocks. You can't do any of this without them. Brain organoids are defined as self-organizing, three-dimensional tissues.
Self-organizing, so you don't build them piece by piece.
Exactly, you don't. They're grown in the laboratory using these highly specialized cells called pluripotent stem cells. The critical feature of these cells is their potential. I mean, they can differentiate or, turn into almost any cell type in the body.
So you give them the right nudge and they become a neuron.
Precisely. When you culture them under just the right chemical and physical conditions, and that process is a whole science in itself, these cells are coaxed to follow a developmental pathway. They self-assemble and they start to mimic the astonishing complexity of early human neurodevelopment.
So they're building structure on their own, just guided by their own biological instructions.
Yes. So they are not just randomly dispersed cells in a soup, they're structurally organized, a lot like actual developing brain tissue.
That's the key. They recapitulate aspects of early human neurodevelopment.
They do. They develop distinct functional layers, which are very similar to the layers you find in the human cortex. And crucially, they form functional synapses.
The connections. The junctions where the signals actually pass between neurons.
The very same. And it goes beyond simple connections. They exhibit collective behavior. They show complex oscillatory dynamics and rhythms. You can actually see the coordinated firing of large groups of neurons.
And that synchronized activity, that sounds like the functional element we're actually trying to tap into. That's what gives them computational utility, right?
Absolutely. The source material captures this essence really beautifully. It describes them as lanterns that let us watch mind-like activity in miniature.
I like that.
It's a functional, albeit very rudimentary, glimpse into how an incredibly complex, high-plasticity neural network processes and retains information. This isn't just tissue, it's a living computational medium.
Okay, so that brings us to the hybrid leap. We have these miniature living networks in a dish. But the challenge is connecting that organic, noisy, wet biological complexity to our predictable dry digital systems.
Yeah.
How do we even begin to engineer that two-way communication?
The interface is everything. It's the whole ball game. We achieve this hybrid leap by directly interfacing the organoids with advanced microelectronics.
What?
Specifically through devices called microelectrode arrays, or MEAs. These are incredibly sophisticated chips embedded with thousands of tiny electrodes that sit either right underneath or directly adjacent to the neural tissue.
So it's listening and talking at the same time.
It is. The engineering breakthrough here is creating a seamless, non-destructive, and high-bandwidth bridge. So the digital components, which include specialized amplifiers and complex signal processors, they interpret the electrical chatter coming from the tissue. And then simultaneously, the digital components can send precisely timed electrical pulses back into the tissue. It's a closed, bi-directional control loop.
So we're essentially, crafting a nervous system for the AI. The AI handles the logic and the I/O, the input-output, but the core memory and the processing that's delegated to the biological substrate.
That is a perfect analogy. We are literally grafting laboratory-grown neural wetware into the body of an AI. The AI becomes the masterful interpreter, the rapid controller, the interface with the outside world. But the actual flexible, low-power learning module is the biological core. It's the thing that can change its mind, so to speak.
And this convergence, this leads directly to the grand vision, organoid intelligence. OI. This isn't just about making faster computers. It seems to be about making smarter, more adaptable ones.
That's the goal.
So why is OI positioned to crack limitations that, billions of dollars spent on conventional silicon, and even these advanced neuromorphic chips haven't been able to solve?
Well, the core value proposition of OI is that it harnesses living circuits that can reorganize and continue learning over time. That is the fundamental non-negotiable difference.
Okay, so let's talk about neuromorphic chips for a second. They try to mimic the brain's structure, right?
They do, but they are still digital circuits at their heart. The weights and the connections they use once they're trained are represented by fixed digital or analog values. They might be energy efficient, sure, but they're still just an approximation of biology.
They're simulations. They're not the thing itself.
Exactly. A trained silicon model, or even one of these specialized neuromorphic chips, has weights that are basically frozen after deployment. They might update slowly with new external data sets, but they lack the capacity for genuine physical restructuring in real time.
But a living neural network.
A living neural network, by contrast, is completely dynamic. Plasticity is its defining characteristic. OI aims to capture this incredible biological adaptability to create computational systems that are inherently more energy efficient and far, far superior at continuous autonomous adaptation in volatile, unpredictable environments.
So it's a paradigm shift.
It shifts the paradigm from computation that merely executes a fixed program to computation that actually evolves its own architecture based on its experience.
Let's pull back the curtain a bit on the engineering. When we talk about a functional hybrid system, we are looking at something far more complicated than just, placing an organoid on a dish of electrodes.
Oh, much more.
We need to detail the 4 required components that form the working engineering blueprint for this really cutting-edge technology.
Right, you can think of it as 4 essential layers that all have to communicate flawlessly. And it all starts with the living heart of the system.
Which is the biological core.
Yes, and this is the high plasticity substrate itself. When we say neurons, we are really only telling half the story. The core includes the complete environment, meaning not just the primary signal processors, the neurons, but also all the crucial supporting cells like astrocytes and glial cells. These cells play a critical role. They regulate the chemical environment, and they're deeply involved in synaptic function and memory formation.
So they're not just filler.
Not at all. This core is the engine of adaptability. Its entire purpose is to literally change its physical connectivity, synaptic plasticity in response to experience. It's the dynamic living memory of the system that dictates whether a learned pattern will persist or, be pruned away.
Okay, so that's layer 1. Moving to the second layer, this is the bridge between the worlds, the electrophysiology layer. This is the hardware that translates the neuron's wet electrical signals into a dry digital data stream.
And this is that microelectrode array, the MEA we mentioned, and the sophistication here is just staggering. We are talking about high-density arrays that are capable of monitoring thousands of individual locations simultaneously. It gives us unparalleled spatiotemporal resolution.
A very detailed picture.
An incredibly detailed picture. And as we said, it serves a dual function. It's the delivery mechanism, sending electrical signals in precise patterns to encode inputs, that's the AI talking to the organoid, and crucially, it's the listener. It records the two key outputs of the network.
And what are those two outputs?
First, you have the individual extracellular spikes. You can think of these as the digital ones and zeros of the biological world. They are the communication pulses between single neurons.
And the second?
The second, and often more revealing for computational purposes, are the local field potentials, or LFPs.
Okay, let's stop on LFPs for a moment. In the original discussion, we described them a bit vaguely as collective synchronized rhythms. Why are these LFP signals so critical to decoding what's going on, to decoding intelligence?
That is a vital distinction to make. A spike is the output of a single neuron. It's one voice shouting. LFPs, on the other hand, represent the aggregated, smooth electrical activity of a whole population of neurons in a specific local area. They are the sum total of all the synaptic input activity.
So it's the murmur of the crowd, not the individual shout.
That's a great way to put it. When the organoid processes information, it's not just one neuron firing. It's a million subtle interactions happening all at once. Decoding OI doesn't rely on tracking every single spike. That would be computationally overwhelming.
Just too much data.
Way too much. Instead, researchers rely on interpreting these collective LFP signals. They often reflect higher-level network states, oscillations, and maybe even cognitive processes like attention or memory consolidation. It's analyzing the collective hum rather than every individual click. And that population dynamic is absolutely essential for closing the control.
Makes perfect sense. It's a measure of the emergent behavior of the network, which is what we're trying to leverage in the first place. Okay, so that leads us to the third component, the stimulation protocols. How does the AI encode an instruction in a way that the biological system not only understands, but also, learns from?
This is where you see a profound mixture of electrical engineering and pharmacology. Inputs can be encoded using extremely precise electrical patterns delivered via the MEA, or in some very specialized setups, through optogenetic stimulation.
Which is using light to control neurons.
Right, where light is used to control genetically modified neurons. But the truly advanced aspect here involves the use of neuromodulators to adjust the learning regimes.
Can you clarify the role of neuromodulators for us? I think most people know neurotransmitters transmit a signal. But what exactly does a neuromodulator do?
That's the critical distinction that's often missed. So a neurotransmitter like glutamate or GABA acts really fast and sends a direct signal from point A to point B. It's the message.
Okay.
A neuromodulator, however, acts over a much broader area, and it changes the context or the modality of the learning. You could say it changes the network's mood.
Like a system-wide setting.
Exactly. Think of them as the chemical switches that determine the network state. For instance, researchers might introduce chemicals that mimic dopamine to reinforce a successful pattern. That effectively signals a reward, which makes those synaptic connections stronger.
I see.
Or they might use chemicals that promote synaptic pruning during a simulated sleep phase. They are using this whole biological toolkit to chemically control whether the network is, say, focused and ready to learn aggressively, or consolidating memory, or inhibiting unwanted activity.
So the AI isn't just sending inputs, it's chemically or electrically determining the organoid's current learning strategy. That is an incredibly sophisticated level of control.
It is. And this sophistication is necessary because we also use activity-dependent feedback. If the organoid's LFP signal shows a desired classification pattern, the system reinforces that dynamic immediately, chemically or electrically, making that connection more probable in the future.
And all of that leads us to the final component, decoding and control. Once the organoid has processed the information and generated a pattern, how does the AI interpret that output and turn it back into usable digital information?
This is the realm of advanced machine learning and digital signal processing. Once the MEA records those millions of spikes and LFP signatures, the digital side has to immediately process it. You need specialized algorithms to filter out all the inherent biological noise and extract the meaningful features from that data stream.
It must be a very noisy signal.
Incredibly noisy. Then, high-speed AI models take over, and they classify the resulting patterns. They're trained to look for specific LFP waveforms or synchronization patterns that correspond to a desired output. For instance, the input was classified as a male voice, or the input is an anomaly. This classification is what closes the control loop.
So without that digital decoder.
Without the digital decoder, the biological response is just noisy electrical activity. The AI is what translates that noise into computation and ultimately actionable intelligence.
Which brings us to the core advantage here, learning dynamics, plasticity versus stasis. This biological power is what makes all that incredible architectural complexity worthwhile.
Absolutely. Living neural networks fundamentally reorganize themselves. I mean, we are talking about synaptic strength shifting through processes like long-term potentiation, LTP, and long-term depression, LTD.
So physical changes.
Real physical chemical changes that make connections stronger or weaker. Neuronal bursts synchronize, and new rhythms entrain to the signals. This is genuine physical change in the structure of the computational substrate itself.
And this is the stark contrast with the digital world.
Precisely. In digital AI, you take a deep neural network, you train it, its weights are optimized, and then they're fixed, they're frozen when you deploy the model. If the environment changes, if the input data shifts significantly, that fixed model becomes brittle and fails.
So you have to start over.
You have to start over with a massive, expensive retraining exercise.
So the organoid systems offer this inherent, continuous low-power adaptability, while silicon offers speed and precision, but requires these frequent energy-intensive interventions just to stay relevant.
That's the key takeaway. The organoid systems, because they're alive and homeostatic, they just keep learning and adapting organically. They offer a mechanism for continuous learning in volatile environments. It is resilience that's built into the design, not added on top with redundant layers.
Right.
You know, if we connect this to the bigger picture, it explains why biology is so fundamentally robust in the natural world. Silicon seeks robustness through redundancy,
Meaning, build more of them.
meaning building multiple, identical, fixed systems, or using complex digital error correction. Biology, on the other hand, finds its robustness primarily through plasticity. If an input changes or a pathway in the network fails, the tissue just reorganizes its internal connections to find a new functional solution. It adapts its own architecture, which is an order of magnitude more sophisticated than simply fixing an error.
This sounds like a future concept, but you're saying the progress is real. Let's ground this in some hard facts. What can hybrids do today? Where is the concrete evidence that this level of biodigital integration is actually functional?
The field is maturing very, very quickly. Hybrid platforms have moved past being theoretical concepts to demonstrating tangible functions, mostly in things like pattern recognition and adaptive signal processing. They're small, but they are operational.
Okay, give us the specific, maybe the headline-grabbing evidence. What has a biocomputer actually succeeded in doing in a controlled environment?
Well, the most emblematic achievement that's been reported involved a system where laboratory-grown human brain tissue was integrated into electronic hardware, and it successfully performed a task requiring some pretty high-level sensory interpretation. It recognized voices.
Voice recognition. That's tough, even for advanced digital systems, because human voices are so inherently variable.
Yeah.
How did the wetware handle a task like that?
It was a beautiful proof of concept. The digital system fed audio samples, which are complex, noisy, variable signals, into the MEA as electrical stimulation patterns. The organoid, acting as the dynamic processor, then learned to recognize the characteristic patterns of certain voices.
So the learning wasn't happening on the chip.
No, the digital component's role wasn't to learn the voices. Its job was to translate the complex audio into a meaningful electrical input, and then translate the resulting LFP activity back into a classified output, voice A or voice B.
No, the core classification.
The core classification, the learning, and the ability to generalize from those complex patterns resided entirely in the organoid's physical reconfigurations.
That is genuinely groundbreaking. It suggests the potential for dynamic, low-power filtering of all kinds of sensory data. Are these systems being given a specific name?
They are. Such systems are often dubbed biocomputers, or specifically in the context of these organoid systems, brain-aware.
Brain-aware.
And the terminology really highlights that this organoid-based learning complements silicon. It doesn't just replace it. It's a mechanism that provides dynamic reconfiguration without the need to scrap the old model and launch a massive, energy-intensive retraining process from scratch.
So how does it handle the noise? I mean, we've established the biological side is chaotic.
Right. And here's where it gets really interesting. To handle the inherent noisiness of that biological data, the digital side of brain-aware often employs things like Bayesian decoding methods or other advanced probabilistic algorithms.
So it's playing the odds.
It's playing the odds, exactly. These algorithms don't look for a fixed, perfect signal. They're designed to find the most probable output from a chaotic input stream. This allows the digital system to manage the biological chaos while still benefiting from its plasticity. The biological learning is low power, self-modifying, and highly tolerant of complex inputs.
That explanation makes the dual architecture so much clearer. We've hinted at it throughout, but let's explicitly address silicon's shortcomings in detail. I mean, if digital AI is driving modern civilization, why this desperate push for complex wetware? What are the three core limits that OI is really positioned to solve?
The current digital AI paradigm, especially when you look at large language models and complex deep learning, is facing some existential challenges related to scale and physics.
Okay, number one.
The first and most critical limit is high energy consumption. Training state-of-the-art AI models requires these vast data centers, and they consume truly staggering amounts of power. We're talking measured in gigawatt hours. A single complex training run for a large model can generate a carbon footprint equivalent to the lifetime emissions of several cars.
That's an astonishing ratio. That's not sustainable.
It's not. Now contrast that with biology. Biological networks, including these organoids, operate at dramatically lower power levels. We're talking near milliwatts, using simple chemical energy like glucose and oxygen. If we can successfully harness even a fraction of that biological efficiency, the energy savings in global computation would be transformative. It would solve the energy bottleneck for the entire industry.
Okay, so that's energy. The second limit relates more to the quality of the intelligence itself, the inability to flexibly learn outside of a very defined box.
That's narrow adaptability. Digital AI systems are superb experts at the specific constrained tasks they were trained on, identifying specific image types, translating specific language pairs. They're brilliant at that. But if you ask that model to pivot to a new, unrelated task, say, going from analyzing MRI scans to designing mechanical parts, it fails completely. It requires extensive and expensive retraining. They are experts, but they are very, very narrow experts. The inherent physical plasticity of biological systems suggests a viable pathway toward more general-purpose, cross-domain learning.
And the third limit is the vulnerability that comes from having a fixed training history. What happens when the world changes?
Exactly. That's brittleness under distribution shifts. A distribution shift is when the input data you see in the real world changes dramatically from the data the system was trained on.
For instance,
a complex factory automation system that was trained only on pristine, well-lit components might fail catastrophically when it's introduced to dirtier components or just different lighting. Because its weights are fixed, it cannot adapt to this new visual distribution on the fly.
but organoid intelligence.
Organoid intelligence, because it is designed to continuously reorganize its synaptic pathways, offers the unique promise of maintaining performance and adapting internally, even when the underlying data distribution changes significantly. It achieves robustness through self-modification.
So if we look at the ideal result then, it's not necessarily about replacing silicon, is it? It's about creating a perfectly optimized division of labor.
That's it, exactly. The combination should lead to low-power, resilient computation that is greater than the sum of its parts. The silicon handles the fast, precise numerical logic, the data retrieval, and the interface management.
And the biological core.
The biological core handles the messy, energy-efficient continuous learning and the complex pattern matching amid all that uncertainty. They are indispensable complements, one for speed and precision, the other for resilience and plasticity.
The potential is clear, but the path forward is, well, it's clearly littered with immense engineering and scientific challenges. We are, at the end of the day, trying to tame life itself. And that variability is the source of the organoid's power, but it must also be a massive engineering headache.
That's absolutely correct.
Yep.
If the goal is large-scale, reproducible, commercially viable organoid intelligence, we have to solve 4 major engineering constraints.
And the first one is surprisingly basic.
It is. The first, and perhaps most immediate, is a problem of basic physiology, vascularization and scale. Organoids, when you just grow them in a standard petri dish, they lack a robust natural blood supply. This is a critical barrier.
Why does that matter for computation, though?
Well, it limits size. Oxygen and nutrients can only diffuse so far through static tissue. Beyond a certain point, and it's usually around 0.5 millimeter to 1 millimeter, the inner core of the organoid becomes starved.
And it dies.
It leads to necrosis, cell death.
So if the organoid can't grow to sufficient complexity, it can't achieve the high cell count needed for the advanced tasks we're talking about.
Precisely. To scale these OI systems to the kind of cellular complexity needed for, say, human-level learning or sophisticated problem-solving, we need breakthroughs in life support. Active research is focused on areas like microfluidics, creating miniature channels and pumps to actively perfuse the tissue with nutrient-rich medium.
So an artificial circulatory system.
In a way, yes, and also biofabrication, which involves 3D printing scaffolds designed with pre-vascularized structures to ensure delivery across larger volumes. Without a scalable, robust life support system, OI will remain constrained to relatively small, short-lived networks.
Okay, so that's a life support problem. The second constraint deals with the chaos inherent in life, signal controllability. How do we ensure that when the AI delivers an input, the biological system produces a reliable, desired output?
Achieving precise, repeatable input-output mappings in living tissue is notoriously difficult. It's not like a silicon circuit where you know exactly how a voltage pulse will travel. Biological connections are probabilistic, they're noisy, they're subject to constant fluctuation. No two organoids, even if they're grown from the very same cell line, will behave exactly alike.
So every single one is unique.
Everyone is unique. Researchers have to spend a tremendous amount of effort on the digital side, building incredibly complex signal processing chains, things like Kalman filters or advanced noise reduction algorithms, just to cope with the inherent biological variability and noise. The initial programming, or maybe better, the training of the wetware, becomes this monumental task of learning to predict the probable output from a chaotic dynamic system.
And the system doesn't just sit still while you're trying to figure it out. That brings us to the third constraint, longevity and maintenance.
It's an issue of temporal instability, or drift. The biological substrates are constantly in flux. Over a period of weeks and months, cells age, connections get pruned, the entire tissue subtly changes its preferred dynamics.
Which means your decoder stops working.
It means that the digital decoder, which learned to perfectly interpret the tissue's responses on day 30, might be completely inaccurate by day 90.
So what's the solution?
It requires a novel one. We need continual adaptation and calibration of the decoders over time. The digital component has to be designed not just to decode the biological output, but to constantly relearn how to interpret the changing biological components. It's a system learning how to interpret another system that is constantly adapting its own language. The maintenance requirement is fundamentally different from fixed silicon maintenance.
And finally, a problem that plagues so many new fields. The problem of the scientific community itself, which is standardization.
Yes. For any field to move from academic novelty to a commercial technology, standardization is absolutely essential. Currently, there is no canonical architecture for OI. The protocols for growing the organoids, the specific nutrient cocktails, the precise timing of differentiation, they vary wildly from lab to lab.
Everyone's doing it their own way.
Everyone. The design of the electrode interfaces, the material the electrodes are made of, the chemical neuromodulators being used, the training paradigms, they're all proprietary or lab-specific. To mature the field, we need agreed-upon standards, shared benchmarks, and common architectures. Until then, every lab is essentially building its own unique biocomputer, which makes replication and comparison incredibly difficult.
That roadmap shows that the current focus isn't just on discovery, it's on rigorous engineering. Let's outline the specific steps detailed in the research roadmap that show where the aggressive scientific effort is currently focused.
The roadmap is focused on refining the infrastructure and the theory. Starting with interfaces, the goal is standardization and integration. We need high-density electrode arrays that are no longer just patches, but systems that can communicate with thousands, maybe even millions, of cells with minimal invasiveness.
And that includes life support.
Yes, this includes better integration of microfluidic perfusion for stable growth and a focus on multimodal stimulation, so combining electrical, optical, and chemical inputs in a controlled, simultaneous way to achieve far finer control over the network's learning state.
The second area seems to be all about measuring the novel performance features of wetware, metrics.
Right, because you can't optimize what you can't measure consistently. Researchers are developing formal, quantitative benchmarks that are specifically tailored for biological computation. This goes way beyond simple metrics like speed. We need metrics for adaptability. How fast and how effectively does the system reconfigure itself to a new task? We need precise energy efficiency metrics relative to the computational output.
And you need to make sure it doesn't forget.
Critically, yes. We need clear metrics for robustness to drift, and most importantly, for long-term learning without catastrophic forgetting.
Catastrophic forgetting is that hallmark problem in digital AI, where Training the model on task B completely wipes out its ability to perform task A.
Exactly. And since the organoid is designed for continuous learning, we need to prove that it can integrate new information without destroying its previous knowledge. Biological memory, theoretically, manages this through selective pruning and strengthening. Proving that OI systems can replicate this without total memory erasure is a very high-value benchmark.
And the last piece of the roadmap.
The last item is theory. We are currently using digital learning concepts like reinforcement and gradients to train a biological system. We need a formal understanding of how these processes actually interact. We need to formalize hybrid learning rules, how biological processes like homeostasis, which is a system's tendency to maintain a stable internal state, and the actions of neuromodulation interact seamlessly with gradient-based decoders, the backbone of modern AI. It's an entirely new field of computational theory bridging biology and computation.
So we have this low-power, continuously adaptive core. What are the practical consequences? So what does this all mean for you, for the listener, in terms of future application? Let's look at the near-term and the long-term real-world impact. These systems aren't going to be general-purpose number crunchers, are they? They excel in specific niches where digital AI struggles. Environments where data is messy, volatile, and continuously shifting.
That's right. And the first major area of impact is in biomedical sensing and diagnostics. Living neural networks possess an inherent adaptive sensitivity that fixed digital sensors often lack, especially when they're dealing with complex biological noise.
Can you give an example?
Sure. Imagine a hybrid sensor designed to detect complex, subtle physiological signals. Not just a simple chemical level, but the subtle dynamic patterns and chemical markers associated with the very early stages of neurodegenerative diseases, like Alzheimer's.
So it's looking for a signature, not a single value.
A signature, exactly. The organoid core, being living human tissue, is optimally tuned to recognize these biological patterns, and the hybrid decoder translates that subtle biological pattern into an actionable digital output. It creates a completely new adaptive class of diagnostic tool.
The second application seems to tie directly back to solving that immense power crisis facing AI. Low-power adaptive edge computing.
This is the killer application, I think. Edge computing means computation performed locally on a device, not sent to a distant cloud server. Think field robotics, long-term environmental sensors, or critical remote monitoring stations.
Places where you can't just plug into the wall.
Precisely. These are environments where energy budgets are sparse, maybe they're battery-powered, and the conditions are constantly changing. A wetware coprocessor could handle tasks like anomaly detection and continuous adaptation autonomously with minimal power draw and without needing to be repeatedly retrained by the cloud. This makes them far more resilient and sustainable than deploying current, power-hungry digital chips in remote or extreme conditions.
And the third area is sort of self-referential, feeding directly back into the scientific quest. Neuroscience in the loop AI.
It's a perfect feedback loop. By coupling high-precision AI to these organoids, we are creating the ultimate controlled experimental testbeds for brain research. We can systematically probe fundamental questions about learning rules, plasticity, and how memory consolidation works, all under closed-loop digital control.
So we can ask it questions.
We can test specific hypotheses about why we forget, or why some memories stick, or how different neuromodulators affect learning all in a functional, human-derived system. This refines both our computational theory of the brain and improves the tools we use to interface with living tissue. It's a virtuous circle of discovery, using AI to understand life and using life to improve AI.
As the capabilities of these brain-aware systems increase, we have to address the ethical horizon. This isn't just philosophical musing anymore, it's a necessary, practical discussion. When does a complex, adaptive biocomputer become something that requires moral consideration?
This is the central, unavoidable question that institutions like the National Institutes of Health and various international bodies are grappling with right now. As these hybrid systems cultivate increasing complexity, responsiveness, and evidence of self-organization, the moral threshold becomes intensely debated.
It's a moving target.
It is, and we have to ask, when do we reach a point where questions of high-level awareness or even basic sentience arise? This requires developing rigorous, observable, and consensus-driven criteria for assessing potential experience, however nascent that experience might be. We need to preemptively determine the line between a highly advanced tool and an entity that requires moral consideration.
And in the meantime, while the philosophers debate that moral line, we need practical, enforceable frameworks today.
Exactly. We need proactive, practical governance. These frameworks must address everything from the origin of the cells ensuring strict donor consent and provenance to setting clear experimental limits. Labs need staged oversight, where the level of ethical review increases dramatically in lockstep with the complexity and the observed responsiveness of the organoid system.
So more complexity, more scrutiny.
Much more. Transparency and clear, internationally agreed-upon boundaries are absolutely essential to ensure that this revolutionary innovation respects human dignity and avoids future harm. We must develop the rules faster than the science can break them.
That's a crucial area of focus. Finally, let's wrap up by exploring some of the highly speculative futures, taking the principles we've discussed and just imagining where this technology might lead in the next few decades.
If those constraints we talked about, vascularization, standardization, signal controllability, if those are solved, the potential applications move into truly fascinating high-stakes territory.
Like what?
Well, one thought is living co-processors in spacecraft. In deep space missions, radiation and unforeseen anomalies are constants. Your silicon handles the high-speed navigation and precise trajectory control.
But the biological part.
But the biological modules are embedded specifically to adapt to noise, radiation damage, and unforeseen microanomalies. Resilience through biological plasticity would be a critical, potentially life-saving feature in environments where human intervention can take years.
I love that idea, the machine having a built-in biological ability to deal with the chaos of deep space.
Another fascinating idea is dreaming machines. Imagine organoid cores that are periodically taken offline and embedded in virtual high-fidelity environments.
Like the matrix for organoids.
Kind of. Similar to how our brains consolidate learning and memory during sleep, the biological core could enter a phase to sleep and process the day's inputs. This biological consolidation process could generate new internal representations, novel solutions, or intuitive leaps that the digital models could then mine and use to solve complex problems. It's leveraging the biological mechanism for creative insight.
So you're suggesting using the efficiency of biological memory consolidation to generate computational novelty? That's incredible.
Precisely. And finally, the ultimate complexity. Ecologies of computation. This moves beyond just a single hybrid system to vast connected networks of hybrid nodes.
A whole ecosystem.
A whole computational ecosystem. Imagine some nodes are specialized for short-term memory, some for complex pattern prediction, and others are simply tasked with rapid learning. These networks would share tasks, and guided by high-level objective functions, some nodes may be instructed to grow new connections, while others prune old, unused ones. You'd be creating an evolving computational ecology designed not just to be smart, but to be a self-sustaining, continuously adapting system that literally manages its own computational resources and capacity. To synthesize this immense deep dive, let's just confirm the progress. Hybrid systems that combine organic neural tissue with AI organoid intelligence. They are transitioning from an academic vision to an engineering reality. They are solving specific core problems that silicon cannot. We have early brain-aware systems performing complex adaptive tasks with voice recognition, demonstrating a viable path to low-power computation that genuinely leverages human plasticity.
And the scientific mandate is clear. We have to solve the immense engineering challenges of scale, control, and standardization. We have to learn how to tame the inherent variability of life without eradicating it, because that variability is the source of its adaptive genius and its efficiency.
That's the tightrope we have to walk.
So here is a final provocative thought for you to consider. If biological plasticity is the ultimate source of computational robustness, and if we are now learning how to graft that fundamental biological power into our machines, what does it truly mean for the future of intelligence, when our systems are designed not just to execute fixed programming, but to continuously learn, restructure, and evolve in response to the world around them? The mystery is the point computation that breathes in circuits that never stop listening.